Today on Surfing the NASH Tsunami.
We have no data showing that NASH resolution on liver biopsy correlates with outcomes. That data does not exist. The FDA accepted this point because they had to come up with something as a path for drug approval.
This weekend, over 200 NASH stakeholders have gathered in Deer Valley, Utah for the 5th NASH-TAG meeting. Join Global Liver Institute President and CEO Donna Cryer and pricing and forecasting guru Roger Green, along with our guest surfers Dr. Jörn Schattenberg of the University of Mainz, Germany, and Dr. Naim Alkhouri of the Arizona Liver Health Institute, as they identify and debate major questions from NASH-TAG Session 4 on the Surfing the NASH Tsunami podcast.
For everyone with an interest in NASH, or more broadly, fatty liver disease, Surf's Up, Season 2, Episode 14, coverage of the final session of NASH-TAG 2021 starts now. So, NASH-TAG wrapped up last night, and the sun rose a little bit earlier today, actually. That's a reference to daylight savings time starting in the U.S. this morning. But the sun rose a little bit earlier because there was a little more brightness in the world of fatty liver disease, he said metaphorically. So most of the people who are in Utah are in various states of leaving or getting ready to ski one more day, as our friend Naim is. We'll talk about that in a second. Louise will not be with us today because she's jabbing vaccines at the Excel Center. But we are fortunate to have Naim Alkhouri joining us before he goes off on the slopes, and Jörn Schattenberg from Germany, and then Donna Cryer. Naim, first of all, I feel compelled to report that I've asked this question already and that you did not ski yesterday. Inquiring minds wanted to know how your first ski session was going to be. We'll let them know that's this morning, and we'll report back on some future episode. Jörn, how was the party with the 6-year-olds?
Yeah, it was great. They managed to find the treasure in the end, and everybody was happy.
Mm-hmm. And the house stayed reasonably well organized, huh?
Yes, it was adapted to the old situation. So it was a small group of 6-year-old girls. I think it could have been worse with boys.
I'll bet on that. And Don, did you get enough rest last night?
I don't know about rest, but we watched some very funny movies, so we had some good family time.
Since there are only 4 of us, I think there's time for a brief and pointed warm-up question. In warm-up, each of you, tell me the one slide in any presentation during the entire meeting that grabbed you the most, and then tell us why. If you want to take a minute, I'll actually go first, because that means no one takes my slide, which was the slide Vlad Ratziu put up in session 3 about doing away entirely with phase 2b trials. I thought that was remarkably bold and made sense from every perspective you could come from: commercial, medical, patient-focused. I just thought it was a tremendous slide about the possibilities of how much we can change if we dare to imagine and then be energetic about the things that we imagine. So that was mine.
I think that was very provocative. I don't think we can skip phase 2b. We can maybe skip a phase 2b with biopsy, but I think, you're still trying to find the best dose. You're still looking at tolerability. So I think, you can do your proof of concept, make sure that you have good target engagement, you see some efficacy. But I think, you still need a phase 2b before you move to a phase 3. Now, can you do a phase 2b without a biopsy? Absolutely. And, Roger, I've been an advocate to try to get biopsy out of the picture completely because I just don't see it as a way for the future. But I think, you still need to go through the steps of 2a, 2b, and then phase 3, because honestly, we don't want to have more failures in phase 3 if we can minimize it by conducting a phase 2b and, again, looking at efficacy in a larger cohort and looking for a safety signal and deciding on the dose before you move to a phase 3.
So I take your point. Putting that in context, there are other diseases. Oncology, notably, has moved away from, maybe even moved away from phase 3 in some cases, where they have a phase 2 and a phase 3 double up in a single study. If we're bold enough, we might figure out ways to collapse things, and 2b without a biopsy is a great start, obviously. Favorite slide from somebody else?
I liked Stephen's presentation on seladelpar just because, to me, it highlighted all these additional features of histology with NASH, and I think it's very clinically relevant. And as I mentioned earlier, I don't want to overreact to a few plasma cells on a biopsy and then get my patients with NASH mistakenly on steroids. I thought that was very eye-opening to me. And then I agree, Vlad's presentation on how we can design trials without biopsy, that was very provocative. I liked his cult slide asking us to believe in a world without a liver biopsy. I think, I have joined this cult already, so I didn't need the encouragement. I am a believer. I do think that we can move away from liver biopsy, but I thought that was very provocative.
I had a really great call recently with all of the Global Liver Institute interns who, in this current time we're able to draw from around the world. And so they're a fascinating mix of medicine and business, double minors or double majors, and some are in classics and public health. And as I pulled Marcus Aurelius' Meditations off the shelf behind me, they were excited and relieved to think that you could combine a classic education with wanting to have a career in serious science. So the slides that resonated with me were when Vlad and Mary, and then even Stephen at the end, uses various works of classical art to illustrate their point, whether it was ugliness of the battlefield and the lessons that we need to learn from all these failed trials, to picture of Icarus, having that pictures of Icarus certainly on what lessons should we draw. Did we fail in construction of trials? Is it that we failed in the path of trials? But certainly to aim high and to be ambitious is not the failure. It's a fantastic message to carry forward. And then the final on Achilles, that just one spot that wasn't dipped in the waters by his goddess mother Thetis is, something for us all to make sure that, we never think that we have the answer so far, so big and so bold that we aren't mindful of our Achilles heel and look for the weak spots in whatever we do. So I loved the use of the classics to illustrate points and remind us of the larger messages and the humanity of the work that everyone is doing.
Absolutely. I'm going to uphold my good friend and colleague from Newcastle, Quentin, who presented on the power of AI to remind us how distinct patients in the same fibrosis category could be. F2 disease is not F2 disease if you look at or even F3, if you look at the type of septa and how they are organized. And I think there is so much that we still have to understand. And that kind of goes down the road that Naim highlighted, where we're going to use integrative models to predict outcome and build new endpoints in these trials. And I think that's a very exciting conclusion for me from this NASH-TAG and something that's emerging in the field, that path away from histology is paved. And we have machine models that support us in integrating different biological information to decide if a patient is benefiting from treatment.
So thanks, Jörn. I think these are all really good points. I also should mention to you folks, since a trick of doing podcasts is that I wrapped up my comments on our session yesterday with no one hearing them at the time. And that episode only went live in the middle of the night, so I'm doubting you've heard them yet. My final comment in our session yesterday was that, to me, the big law of unintended consequences, and this goes back to all this, is that we're going to have drugs in 2 years. And in Europe, it may be that if we have drugs and the label says biopsy, patients have to get biopsies. But that won't be the case in the U.S. It'll be the payers who drive, and the payers will drive in different directions. So if we're not prepared to think about what the world after biopsy looks like in terms of real patient treatment, and I think the agency will wind up getting dragged along faster in different directions than they expect by the decisions that are made once they're actual patients. That's a moment I think we'll have to be ready for.
I couldn't agree more. I think we need to be ready for, drug approval, and we need to have a plan to avoid, the payers asking us for a biopsy as a hurdle to deny patients treatment. I think we have enough data now from non-invasive tests to identify what we call at-risk or high-risk NASH patients without a biopsy. In fact, if you have 2 non-invasive tests that are agreeing that the patient is high risk, the chances of them having high-risk disease on biopsy, it's in the 90% range. So I really don't need to biopsy these patients. And it will be really a sad situation for, the NASH community if we are required to biopsy them. Now, the other argument that I hear a lot is, well, yes, these NITs are good at baseline, but we still don't have enough data to monitor treatment with them. And we need something that correlates with outcome. And the point I tried to make yesterday was that, we have no data showing that NASH resolution on liver biopsy correlates with outcomes. That data does not exist. The FDA accepted this point because they had to come up with something as a path for drug approval. And we need to be aware of this. If insurance is going to tell me that ballooning resolution on biopsy is going to predict outcomes 10 years down the line, that's not the case. I mean, the argument that people make is that when you have NASH resolution, this should lead to fibrosis improvement. And then we know that fibrosis stage correlates with outcomes. But, the concept of just NASH resolution by itself predicting outcome is not well validated. This is important to know. So I think, if I have a patient patient with elevated liver stiffness on FibroScan, with elevated ALT, with elevated ELF score at baseline, and then I treat them and I see the stiffness is down, ALT down, ELF is down. That's enough for me. I mean, yes, we still need more validation on the cutoffs to decide on futility, but I think we do not need to do the biopsy, and the outcomes on biopsy, knowing all the limitations for biopsy, even the outcomes are not well validated. So these are, important things to keep in mind, and hopefully we can make these arguments to payers and insurance companies to avoid, the necessity for biopsy, because I worry it's not going to be just biopsy at baseline. Then they're going to require biopsy a year later to see if you're responding or not. And I mean, biopsy comes with risks. And in my mind, I think, what I see in papers on the risk of biopsy is always an underestimate, because people don't want to share the complications they have always. Okay, I'm not saying anyone is trying not to be transparent, but this is the nature of medicine. I mean, things happen, you don't always report on it. I wish we had a registry for liver biopsies to talk about every bad outcome that happens in the real world.
Naim, I really appreciate that, and Roger, and I agree, of course, with that risk to the field of biopsy being the rate limiting factor when we do truly want to scale. For several years, as we've constructed the Global Liver Institute, we're only 6 years old, and we've been having a focus on NASH for 3 of them, or I guess since 2017, almost 4. And there's been pushback as people expect, well, you're the patient organization, so you should be funding research. And I said, well, there's a lot of research going on. I want to make sure that research is pulled through into adoption, or, you're a patient advocacy organization, so you must do support groups. Well, we are starting to put together a support model, but always looking at the field and looking at the, true advocacy gaps and the true risks for the field. We've always focused on this. What would the, value assessment look like, and how do we prove the value of NASH therapies when they arrive? What would the insurance look like, not just for the drug, but for the imaging and all the parts of care that NASH patients would look like? That's also how we got involved in pushing for new ICD-10 coding, so that we would even know and be able to distinguish in the EHR early fibrosis from late fibrosis in a better way, so that we could be targeting things well. And, and then from a policy level, whether it's with FDA or CDC or even with NIH, and what legislation is necessary and built into various vehicles, and would there be champions if we did get constrained by any of these stakeholders? Would we have champions in Congress who understand NASH and are ready for the issues? So we've been, sort of tilling that soil for a long time for the seeds to be planted. When people would ask, well, how does the Global Liver Institute work vis-a-vis the Liver Forum? I said, of which I love and I'm on the steering committee for. And I would always say, well, they're sort of like obstetrics and we're pediatrics. So they're making sure that there's like a healthy baby that is being developed. But once that baby is born, we're going to be there to catch it and receive it and make sure it's raised well. And so, once something in NASH is born, it really is all the parts of, that the Global Liver Institute has been working on, even our awareness of International NASH Day, to make sure that, it's received and raised well and reaches its potential. So all of these issues that people are now starting to really recognize will be important once a drug is approved are the things that we've been working on for several years. And I'm really excited about, the opportunities to rally more people and have more engagement. The policy work group was already standing room only back when we could stand in rooms, and it has been very vibrant throughout, but we need, our other work groups as well need to have that same momentum as we are able to work with communities and payers and policymakers moving forward in one voice and one community and one, focus.
So let me throw out one more digressive thought, and then we'll move forward to talking about yesterday's session, which is that to some degree, I think we're always fighting the last war, and the context in which we're thinking about biopsy and payers is, I think, a context driven by Hep C and by the Gilead drugs and the fuss around that. What I can tell you is, because I did a lot of research on this, a lot of marketing research, a lot of talking to payers at that point in time, and on this issue too, state directors were literally claiming that the Gilead drugs were going to put them out of business. We're going to soak up more than the size of their entire drug budgets. Because remember, the drug came out, it was $96,000 a year in the States. And huge barriers were constructed, I think unnecessarily, but in panic mode, but were constructed based on a panic around that particular dynamic. I haven't heard that panic around NASH drugs for the last year or two. You did 3 or 4 years ago, but you really don't now. And I'm hopeful as a result that payers will be more holistic in how they approach the question because they're not going to feel at the same level of economic jeopardy. Donna, you made a face. I could be completely off base on that. You're spending more time on this issue than I am, but that at least is my hope.
Well, I think that nobody paid that sticker price. So that number, was nobody's actual number. But I think the reaction to it, you're absolutely accurate at what the visceral reaction was that. But working with several in the NASH space, I think particularly at first they did have that case in their head. I think the very important distinctions that we've been making, and I think drawing it back to this meeting where we talk about making sure that we understand and risk stratify and select our patient profiles appropriately and determine who would benefit from drug therapy. And then as we get better to understand which drug, and I think in which combination we'll have productive conversations with payers that will provide access to the appropriate patients for therapies. We've been talking with them now to make sure that they understand the different aspects of weight loss support that should be provided for patients, and seeing if there are any barriers to any type of screening of technology that people need to be identified as those non-invasive diagnostics get better. They're very open to more evidence about how specifically they use different technologies for what purposes. They want clarity around that. And then I think that we can make a very good case for full access to patients, particularly, F3s and F4 compensated, and then certainly decompensated cirrhotics, for whom savings to the system, as well as the clear ROI to the patient, are very clear. What's less easy is to make the economic argument for patients who aren't in the system right now. They're not costing the system anywhere because they're not identified. They're not getting care. And so I remember walking through not the Arizona desert, but the New Mexico desert with some economists asking, well, how do you do better than zero? Because right now, many of our patients are not receiving care, or they're receiving care, but they're not coded as fatty liver disease or NASH, so we're not capturing those costs to be able to make the economic case that providing services to them, would be cost effective. So that type of work, I know Maz has a fantastic paper on cost effectiveness of treating diabetics, and so we need more of that type of work to be able to bring to the value assessment folks and the payers. But that way has been paved, and they're ready to receive that information.
Yeah, I agree. I think it's working. I talked about this, I think, at NASH-TAG in 2020, that in 2019, when you went to payers, Naim, they were willing to throw up any barrier that they could to manage what they thought were going to be these hellacious costs based on being swamped with all NASH patients at once and drugs that cost $20,000 a year and didn't work very well. And between 2019 and 2020, folks had become aware how you could gradate the population in ways they hadn't thought about, and the jawboning on the prices of the drugs had come down, and payers just seemed a lot. And Donna, I think people like GLI were making better arguments, and payers were just becoming more receptive to the idea that you didn't have to view this with the level of terror that they initially approached Hep C.
It seemed from last night, one of my takeaways was that we need to make sure that, genetic testing is reimbursed, is available. So as we start bringing in genetic risk factors for the individual patient and first-degree family members, as was explained, that, I've added that to the list for my staff to make sure that we're looking at access to genetic testing and genetic information outside of clinical research environment. It's an issue that we have stayed active in as a member of the Personalized Medicine Coalition, where I used to be on the board. So I do know, Foundation One. I do know Michael Polini, who's the founder of that, and the lessons that we can learn from, oncology, genomics. So we want to make sure that our patients have access, if that proves to be the case, that they have access to genetic information as well.
So that's a great bridge to last night. And let's make sure that we don't lose track of last night in the context of everything else we're talking about right now, which is all great stuff. But so Naim, we're going to use the same format we've been using. And as the one person on the call who is in Utah, you draw the straw of going first. The three questions: one point or issue you heard during the second session yesterday, the afternoon session, that you found novel, surprising, or likely to drive a different course of action. And then what the presentations in the session had to say about that issue.
We had basic science session, and then we had the new drugs and development session. And honestly, there was so much new information. So I'm just going to give you a quick summary on the points that I thought they were interesting. I think, to Donna Donna's point, genetic testing is becoming cheaper and cheaper. In hepatology today, we have what we call the cholestasis panel that can screen for several genes for liver diseases that can cause cholestasis. And we are learning more and more about different polymorphisms in NASH, mainly PNPLA3, but also TM6SF2 and MBOT7. And I think, in the future, once we understand the effects of these polymorphisms on disease progression, risk of liver cancer, and also have more personalized medications, things that Dr. Lumba mentioned in his talk, where you have these antisense oligonucleotides, short interfering RNA, where you can target one specific gene and its expression. It's going to be very important for us to have these genetic panels available, and potentially they could be supported by pharma companies. And this is the case with the cholestasis panel. I mean, I have these kits in my office and I do them for free for my patients. You can test for several genes for cholestasis. I would love to have the same paradigm in NASH and, try to understand on a more personalized level what's driving the disease in each patient I see in clinic. So that was Dr. Lumba's presentation. We heard from Dr. Schnabel about the role of microbiome, and he mentioned very fascinating facts related to the topic, including the fact that the gut microbiota, even in children, can produce what we call endogenous alcohols, and the potential that these endogenous alcohols are also drivers of NASH and disease progression. He also mentioned a couple of very fascinating concepts of kind of like affecting the microbiome with the concept called drug the bug, where you can actually use viruses, or what we call phages, to kill the kind of bad bacteria and bring up the good bacteria population. That could be done also potentially with antibiotics. And then he mentioned another concept, which is drugs from bugs, where you can actually potentially have some of the products that are produced in the gut bacteria as therapeutics. And one example he gave was short-chain fatty acids. And then we heard from Dr. Friedman about fibrosis targeting and looking at the TGF-beta pathway and using integrins as a more selective way to target the TGF-beta pathway. Dr. Friedman mentioned few companies in his presentation, and he is still optimistic about anti-fibrotics and the fact that they will play a role. We also had a brief discussion on potentially combination therapy, where you have an anti-steatosis agent combined with an anti-fibrotic agent, so you can target upstream and downstream. Then we heard from Dr. Tetri on the use of preclinical models and some of the issues, and he discussed a few of the success stories and failure stories where the animal model data did not translate into human success. And I think one thing that's emerging is that probably you need more than one animal model to validate your drug before you move it into human studies. I think that will give you more confidence that the effect you see is not limited to just one animal model, because unfortunately none of the animal models capture all the features of the human disease. We tend to also test younger animals, so one of the questions was, are we looking at basically the equivalent of adolescence in human when we are, treating these mice at an earlier age? So I learned a lot in the basic science session, very interactive, and many questions from the audience. I'll stop here and happy to comment on the new drugs, but I'll give a chance for others to comment.
Naim, thank you for summarizing that brilliantly and making highlights. As somebody who's trained in the lab in New York, of course, my heart beats for those animal models, but I also know that there are a lot of animal models that you can, look at the wrong way. And I think that's a very important finding. There are some out there still using type 1 diabetes to study NASH, which I think is so important to keep in mind when you try to interpret your data. So you really got to know those models well before drawing final conclusions on the effectivity of your compound in that model. And I totally agree with you that you should look from different models to see how your compound fits in there. So I think that was a very important session. I would not want to give up on those basic science aspects in NASH, because we're learning so much. And moving on to the pharma, new combinations, new trials, I think it's very interesting to see that some components are re-emerging. Not everything that was on the list is totally new. There's been VAPs by different companies, for example, the VAP1 that have actually been terminated. And now we're seeing another combination emerge here. I do get the feeling that we're still going a little stronger towards combination, looking at the GLP-1 glucagon agonists. I think there's a lot of very exciting data in the pipeline. And, bottom line, I think all these new compounds will benefit from the discussions around biopsies, because they're well advised to really include include a large biomarker panel to give the best information that's available and setting up a trial that could augment biopsy as an endpoint and come up with data that, as we've discussed here previously, is really moving beyond the biopsy. And then just a last quick comment on genetics. I think that introduces a whole new set of complexity in the field because it will give us subgroups that we might have to look at differently. And there's some data out there, for example, that patients with PNPLA3 might be more responsive to lifestyle compared to others. So I think it widens the horizon in terms of who's a responder, who should be treated how, and personalized medicine, of course, is a good way to summarize that. And I guess I'll stop here for a second.
Before we go on to Donna and me, I've got the same question for both of you, which is part of the point of the question is to ask what is the one thing you might do differently next week or next month as a result of what you heard in the session. So Jörn, let me ask you that first, and then Naim, and then we'll go on to Donna's observations and mine.
I guess I'll do more genotyping. I'm really interested to learn more about that. And I do have a cholestasis panel in my unexplained LFT workups. I haven't used it as routinely in my NASH patients, but I think it's a good way to subsegment that patient population and provide additional information also from the patient perspective and guide them.
Naim, one thing you might do differently next week, next month as a result.
One thing we learned from the microbiome talk that, using antibiotics, antifungals, changing the gut microbiome may have a profound impact on NASH. And, it got me thinking, and it was also a question from the audience, that how important it is to, look at these patients that receive antibiotics during the context of a short clinical trial and actually maybe think about analyzing them separately if it ends up being a substantial cohort within a clinical trial. I mean, we always have to report every medication they go on during the context of a trial, but I don't think we pay too much attention to a short course of antibiotics. I think it's important to potentially analyze this data and maybe, you can't really come out with any conclusions from one trial, but if you look at maybe combining data from several trials and analyzing, people that receive antibiotics versus those that don't, that could be interesting concept. I mean, I'm not saying it's going to make a big difference, but I would like to know. I would like to look into this a little bit more. So moving forward, I think I'm going to take antibiotics more seriously in the context of, taking care of patients with NASH and NASH clinical trials.
Donna, let's move on to your big aha from yesterday afternoon.
Well, if I was a person who was inclined to have a glass of wine after several hours of sessions, I was definitely dissuaded by several of the presentations, whether it's, effect on potential for fat accumulation in my liver or just upsetting my delicate microbiome. So, it was the tonic and lime without the gin last night for me. So I think people were quite persuasive. And as I think about this and listen sort of with my patient ear, what would the average person, how would they apply even these early learnings and signals from the session? So the NIH and the new nutrition guidelines, came out and at first wanted to advise that people only have one drink a day, even for men. And there was so much pushback on that. But, it really looks like the data is that, no amount of alcohol may be safe, particularly with someone for some version of liver disease. So I think as an average person who cares about their liver health and may even have some even early stage, let alone late stage, I would definitely be advising people, well, no alcohol, buy a scale so you can assess your lean body mass. Don't worry so much about your BMI, but let's figure out what your body composition is so you can communicate that with your doctor. I don't have clear guidance on nutrition yet, although we can say it's sort of Mediterranean in a way, but whatever it is, you need to be supported in your nutrition. And I think as we as a field need to figure out what nutrition recommendations and lifestyle we would want to standardize and that really works for NASH patients. I'd want to have genetic testing for me and my family to understand, a more personalized risk. And I would be far more fascinated about microbiome testing. And even, I love that we just now refer to it as, FMT. It doesn't sound as gross, that way. And looking at the increases in safety of, fecal myobial transplant, now that it's, pooled stool and in capsules and things like that, I would be more interested as a patient to explore that. Might bring up the issue of antibiotics to my physician and particularly be more forthcoming if, I've been on them or, I've taken some just because they were in the, box on the shelf or something like that. Or if I take probiotics, I would definitely be more forward with my physicians about my use of them so that can be considered. I would certainly be pushing back on biopsy. If my doctor asked me for one, I'd ask him to justify it and ask if there's a non-invasive test that could be used instead. So that's how I would sort of think through some of the applications, in real time for people as we continue to refine and confirm some of these answers.
True confession, I missed parts of the session because of some things that were going on personally here. But I did catch the early parts. I caught Dr. Schabel, and one of the thoughts I had listening to that is, for all the profusion of diet programs of different kinds that are going on, whether you're talking about the people who actually package food out or just the people who do behavioral support, I'm not familiar that any of those focuses on microbiome issues. I mean, they focus on calorie intake and they focus on calorie-dense foods and foods that are likely to do a better job of satiating your appetite. But I'm not familiar that any of the commercial programs right now talk about microbiome in any meaningful way. And if they don't, that's a big miss.
There are several wellness companies and, selling you, microbiome tests and purporting to create personalized supplements for you. I don't know the validity or credibility of those claims or that work, but I think it is worth validating and seeing if it can be integrated. Certainly it's a ripe area for research. And I think it probably is, the reason why some people lose weight on a low-carb diet while some can lose on a relatively high-carb diet, and why people have just sort of thrown their hands up and been like, well, use whatever works and causes weight loss. I think we can start being a little more prescriptive if we understand people's microbiome and can better match patients and understanding this type of diet will be more likely to work for you because of the microbiome you have, rather than just a lot of trial and error and saying, well, just fewer calories or something like that, which is certainly not the answer for somebody who has different metabolic disorders going on.
Donna, when you mention that, those companies that offer personalized approaches, I'm always worried as a physician that the patient puts the wrong focus on, he's distracted about things that might have some evidence. I don't generally advise against that, but I always caution to not lose the main focus of something that has been evidence-based shown. And I think, as you highlighted, there needs to be evidence generated. And I think in the liver field, there's a lot, in the absence of other therapeutics, I feel that patients always ask, what can I do? What type of diet? What's this? Can I buy this supplement? What about? They want to do something for the liver. And I see the risk of commercialization of that field.
Vast risk in the absence of data. So your interesting question, though, is as data is being developed, how quickly can it be constructively commercialized? Because if you Google liver health, you get a whole bunch of consumer ads at the top about different forms of liver detox, none of which are proven to do anything but make people money, as best I can tell, and don't have a tremendous scientific base behind them. As we start to develop scientific foundation on these things, the question is going to be, where is the balance between, if people want to do something, give them something better to do, and have enough proof in what you're about to recommend that they do, that you can have some confidence that you're actually doing good. You know what I mean?
Yeah, and if they Google liver health, the Global Liver Health Institute should be coming up first, and somebody should talk to Donna's team, I guess, and get some advice there.
We're going to work on some SEO and some AdWord purchases, perhaps.
Donna has great SEO people. I'm not worried about that. I'm actually more worried about how much money these other clowns are spending on the bad ideas, but that's neither here nor there. What else about yesterday afternoon session do we want to touch on? I guess I'm also struck by the idea about drugs that come back around again, and drugs that wound up being purposed for one thing that have come back into fatty liver disease and look pretty promising. The CRE431, the Hepion product, for example, was not developed originally for NASH, but appears, I think, reasonably promising here. Tesamorelin wasn't originally developed, obviously, for NASH, but appeared might be promising here. What else do we want to touch on about yesterday before we go to final question and out?
I mean, I can mention a few things about the new drugs. I mean, we had several presentations, including from Pfizer, about their DGAT2 and ACC inhibitors, and they have, two medications now in phase 2B biopsy trial. DGAT2 inhibitors, I mean, it's looking promising, and it seems like it mitigates that increase in triglycerides caused by the ACC. In fact, the ACC inhibitor caused significant elevation in triglycerides by itself, that they're not going to look at it as monotherapy. So it's being developed as combination therapy with DGAT2. So that's an interesting concept, that you can actually look at a drug only as part of combination therapy as treatment for NASH. You don't have to validate it by itself. The VAP1 with turns, I mean, I thought, that was interesting. They highlighted some differences between that one and BI VAP1 inhibitor that was discontinued. There was some data on TLR signaling inhibition, specifically TLR4 with mosedipimod, and they showed some animal data, and it works as an immune modulator to remove these what we call pathogen-associated molecular patterns and damage-associated molecular patterns. These are kind of like, in a simplified way, molecules that the immune system uses to realize that there is danger and that we need to be active. But sometimes overactivation of the immune system is not a good thing. And then, as you mentioned, Roger, the CRV431 with the Hepion, a cyclophilin inhibitor, it has, in my mind, potentially pleiotropic effects on inflammation, fibrosis, and hepatocyte death through the different cyclophilins, A, B, and D. And they actually have tested this in several animal models, so I was impressed. I think in the slide they showed 7 different animal models, and they have a phase 2A program and planning for a phase 2B. And then Altimmune presented some data on their GLP-1 and glucagon receptor dual agonist. And I think, the major difference between this one and medicine like semaglutide, that with semaglutide, which is purely a GLP-1 agonist, we think all the benefits in NASH are driven by weight loss, which is very important, but it doesn't have any direct hepatic effects. By adding this glucagon piece, you can, in a way, defat the liver through mobilization of liver fat, increasing hepatic lipolysis, and these are direct liver effects. And then they compared, their compound to other compounds that are kind of this combination, GLP-1, glucagon, GIP. So I thought that was interesting. So this is my take on the new drugs that were presented.
Nice summary.
Yesterday.
Thank you, Naim. And I think if we are frank, and we've discussed a lot about non-biopsy driven trials, I'd like to revisit the concept and highlight that there's also variability in the biomarkers, of course, but it'll be much easier to get repetitive biomarkers sampling and then kind of build, let's say, baseline value of a patient. And something that was discussed briefly during the entire meeting was, again, the lead-in period. And I'd like to get your thoughts, maybe if we have 5 minutes just on that, and saying, how can we get stabilized patients into a trial, get them metabolically, liver disease at the same level, at their baseline level, not having them going up and down. So I think in the concept of developing biomarkers forward as an endpoint, I'd like to revisit that lead-in period to gather more information on the patient. And maybe one of those compounds you've just mentioned is capable to do that in the future.
Jörn, this concept makes me worry a little bit just, in the context of clinical trials, because, the disease is, it has its own course and ups and downs. And if you think you can stabilize ALT within a 6-week or 8-week, 12-week period, stabilize liver stiffness on FibroScan, it just, it's going to translate into many, many screen failures in clinical trials. Although I like the concept, I like, the idea of kind of like stabilizing everything before you get them on treatment. I feel like we do this in a way because they have to be stable on their diabetes medications, typically for 3 to 6 months. They have to be with a stable weight, typically no more than 5% weight loss before they enter in a trial. I just worry it's going to backfire. I think scientifically it's a sound concept. I think in real life it might be an issue. And then I worry that, the same thing will be used by payers to say, okay, well, you need to do this lifestyle intervention before you qualify for anything, and they decide on the duration. And I think lifestyle is always the best choice. But, if you have someone who had type 2 diabetes for 10, 15 years, they've been told by several doctors, you need to lose weight, you need to do this exercise, and yet they haven't been successful. Is it really going to make a difference now that the liver specialist is telling them they need to lose weight and exercise? In my mind, I don't think so. But I agree. I think, if you have a young patient who has never known that they have metabolic risk factors, they're identified through a screening program, and now we tell them, okay, let's do all these things. Sometimes you see a huge impact. Again, I just, for me, being heavily involved in clinical trials, it just makes me pause a little bit. It makes me worry if we're going to start doing this lead-in phase as a standard in clinical trials, because I feel like we kind of do it already by requiring all the stability features to be present before we enroll them in a trial, and I just don't want to delay enrollment.
Interesting you should come up. Sometimes the best conversations in these things happen after you turn off the recording. Yesterday afternoon, at the end of the session, at the end of the first session, Ian Rowe and I got into a bit of a debate debate about what the meaning of Michael Charlton's Babe Ruth analogy really was. So Ian stuck around after we turned off the tape yesterday, and Donna, right after you dropped off, Ian asked Michael if he had a couple more minutes to answer one more question, and he asked him about the Babe Ruth analogy, and was because we disagreed about it, and could Michael go into more detail. The answer was entirely about Leadin, and it was about Leadin and diet. And his point was that if there are a few dietary factors that are so strongly predictive of how patients might respond, then why don't you lead in with those to wash out your placebo rates? Because if people, what he was trying to manage for was the variability in placebo rate specifically. And his feeling was that if you manage diet as well as the other things that you're managing on the way in and get people pointed more towards doing some good things and not doing some bad things, that the net result would be that you could affect placebo rates on clinical trials, and that might take a little more time at the beginning of trial, but it would improve the comparability of different trial results and the stability of placebo rate. His contention further was that tends to be done already in some centers, but not in others. So that in fact, there may be inherent bias right now by where you go to get your trials done in terms of how your placebo rates are going to be. So that was really, I think, the issue he was staring at. I understand so I'd love for you to comment on that, given the potential downsides that you just mentioned. For both you and Jörn and Donna, all three of you to comment on that.
Well, I've commented because before I heard that, that's how I was interpreting it. I think it is important because I think standardizing that lead-in period, that stabilization process, whatever you want to call it, that it is in fact because so few people have access to care or high quality care, whether in the US or around the world, that it does end up being an intervention of some sort, just that initial contact and the process to prepare to participate in a trial. And so I think we need to bring more rigor and standardization to what we are doing so that we can understand what the effect of it is and is not.
Yeah, and all points are well taken. And I agree with Naim that what are you going to do about patients that go up and down if you have a lead-in and you do repeated biomarker testing? What's your baseline? Are you going to take a median? You're going to take the highest? It makes things more complicated. The comment that the direction it comes from is that you want a homogenized patient population at start, and to decrease placebo response is I think this could be one way to be more successful there. I'm not so worried about the differences between trial centers because that's why, at least in the phase 3, you really go multi-center, multinational. I mean, talking about nutritional differences in Asia and US, I mean, there's a lot of variability already on that level in there. In the end, you hope that it really is the disease phenotype that drives it and the drug that impacts on the primary endpoint by combining so many patients. So, it's just a thought I had, and I agree. I don't want to be interpreting 3 ALT values over a period of 12 weeks to decide if a patient goes on drug or not. It might be worth to educate patients up front to get some of the deleterious nutrients out of their diet that could impact results.
Yeah, to me, it's a good concept that will have bad effects. And I think the same thing can be said about the whole consensus pathology reading and having more pathologists interpret the biopsy. I think it makes sense, but at the end of the day, what happens in real life is the loudest voice in the room will get their way, and then the other pathologists will agree, and liver biopsy is still not a good thing. So I think, I'm being very vocal here. I'm being controversial. I don't mean to offend anyone, but this is my take. And I think the best way is to test it in real life. And, if you have believers in this, let's do it and see what happens to screen failure rate, trial enrollment, and see if it affects the placebo response rate. Because sometimes, you can speculate all you want until you have actual data. I don't think we can make any conclusions. And maybe some companies will choose to go this route to minimize the placebo effect rate. In my mind, if you have a good drug, similar to like the FGF21 data now we saw with Akiro 89 Bio, the semaglutide data, that the placebo effect is not going to kill your drug. I mean, we've seen it with other less potent drugs, but I mean, we do have a problem with the placebo response rate. I would like to again have a scientific answer to this. I think, again, from a scientific point of view, I'm intrigued. From a real life point of view, having to enroll patients in trials every day, it gives me a little bit of a chest pain to hear these ideas.
Well, trying to save everybody's cardiovascular health in as many ways as possible. My sense was, at least in the Babe Ruth conversation, the issue wasn't about judging between 3 ALTs, but just stabilizing everybody's diets and then accepting the variability that would come after you did that. I think when you get to, we're going going to try to decide which measures people take, decide whether they get in the trial or not. Jörn, I'm with you. Now we're back to a lot of human judgment, and that goes along with the consensus pathologists, and we're trying to minimize variability around human judgment here, not maximize it, and I think whatever path we take. So this is the funny thing. I started by asking the question we usually ask at the end, and I think we're going to end by talking about the first thing that came up in the conference, which was Babe Ruth. If anybody has anything else they want to add that they'd like to share with podcast audiences, this is the last thing they're going to hear from us while we're at the conference. So I'll go around and ask everybody if there's any further comments, and if not, we'll just wrap up and send Naim off to breakfast and skiing and everyone else off to their day. Further comments, Naim?
No, I mean, I was encouraged to hear about non-invasive tests and especially the data on how they change in response to therapeutic interventions. I think it's another step to get us closer to avoiding histologic assessment for, response to treatment. So I just wanted to say that. I mean, there are really nice presentations by Dr. Ranella on the serum biomarkers, by Dr. Lumba on imaging biomarkers. I was very intrigued with the platform protocols, the basket trials, umbrella trials. It was a rich meeting overall. I mean, I learned a lot, and I look forward to next year NASH-TAG.
Yeah, absolutely. And I like the discussion around the academic and scientific value of those trials. And I'm supportive. And I guess to go back to Babe Ruth, I'm sure he missed a strike every once in a while, and you just have to have him come up to base frequently enough and get your numbers and your statistics right, and then see if your drug hits the target.
Yeah, I think my takeaway from both the analogy and the conference is to just keep coming up to base and for pitchers to just keep pitching. There's an incredible amount of hope that people should take away from the number of compounds still in the pipeline, the number of approaches and mechanisms that are being tested and developed, and both the humility and curiosity in which the researchers in the field are approaching NASH in a very holistic fashion, borrowing ideas and best practices from oncology or in infectious disease or other areas. And so I think the message is one of really great hope and excitement for the field.
Agreed with all that. And I want to bring it around. We set up this particular form of coverage for the conference to try to give people who couldn't be there a slight flavor of the dialogues and conversations that happen when people aren't in session and are kicking around the issues that are around the session. And kudos to everyone who's been on this podcast. I think generally we've given folks a pretty good flavor. I made this comment a bunch of times. I became enchanted with the whole issue of liver disease attending NASH-TAG in 2019 and 2020. And part of my takeaway is that I think the organizers did a very nice job of pulling this off as a hybrid conference, but I think all of us can't wait until more of us can be back together again next year. I was fascinated by Stephen's comments at the beginning of the first day, and Naim's as well or second session, I guess it was, about what it felt like to be back with your colleagues after 14 months away and what a different setting it was. So my hope is that we can all be together in Park City next January 6th. It's on my calendar already. And that we can continue this dialogue and find more ways to share it with more people outside the room itself, but get more of us into the room as well. With that, let us sign off. Let me thank Naim for getting up at this ridiculous hour, go to breakfast and go skiing. Have a good day. And Donna and Jörn for the hour was not ridiculous, but it is the middle of a Sunday afternoon as well.
This concludes the Surfing the NASH Tsunami coverage of NASH-TAG 2021. Have any questions for the surfers? You can post them on the Surfing the NASH Tsunami discussion group on LinkedIn or Facebook, or send them to questions@surfingnash.com, and we will address them as part of the NASH-TAG wrap-up episode. Thanks for listening. See you Wednesday night at 11 o'clock p.m. Eastern Time for our NASH-TAG wrap-up session on Surfing the NASH Tsunami.